Good morning, good evening, thanks for listening. This is a show on organic gardening, horticulture and agriculture, focusing on plant science, soil science and microbiology. Information is provided for scientific and educational purposes. It's fact-checked. Many of the websites and papers discussed are put in the description below. If you're rewatching it back, fast forward until you see the screen change, unless you like listening to me ramble, because I don't know what today's topic is yet. I got to wait until chat arrives and we go through some stuff in a few minutes and see what discussion happens. And if it's only gone for 10 minutes, there's a short show. So I'll give it till 2 minutes, another 70 seconds. So another 50 seconds to go. Thirty more seconds to go. And if you're listening, put something into chat if you can and see what sort of discussion we can get started on plant science, soil science and microbiology, please. 15 seconds. All right, what can I talk about? Well, there's 2 minutes, so I've got to start and talk about something about plant science, soil science, and microbiology. Well, all I can think about is drought stuff, because that's what's happening. Last month, I got 0 rain. 0. The month before, I got 0.4 mils. So that's sort of high on my agenda. So I've got this cool slide to show. So I've been going on about drought and stuff, and you know I'm into microbes. Let's just see how the microbes interact with the plant's roots. This is the interplay between drought-induced plant exudates that come out from the roots and microbial metabolites that are in the rhizosphere here. So see down here the dots, and it's expanded. So this on the left, it expands. This is the root tip. You can see it there, the quiescent center right in the middle, the root hairs. And what's happened is the root tip releases metabolites, secondary metabolites, like all these ones around the outside. And they're involved in the maintenance and the recruitment of drought-adapted microbial communities. And on the right is the microbial communities. We can see different genus they're from: Rhizosphere, Pseudomonas, Bacillus, Streptomyces, Flavobacterium. And they have an interaction through the rhizosphagy cycle. You know, it goes out through the root hairs and in through the tip. That's one of the cycles. And it's very close contact with the plant's roots. They respond by producing all these different metabolites, which again, in turn, enhance plants tolerant to drought. And this demonstrates the cooperative, symbiotic relationship that happens in drought adaptation. So that's pretty cool. So by having a good assortment of bacteria, they will help you in the drought situations as well. Right next to the rhizoplane, next to the roots. Cool as. I'll put this link up here from Trends in Plant Science, those who are interested later. Hello, Genetic Memory Farms. He says, good evening, Aussie. How are you? Getting ready for the cold. I didn't know what else to speak about, so I was just sharing some more drought stuff because it's last month I officially got 0 rain. But just like those plants' roots, plants' exudates, and the microbes, I've also adapted with the drought situation with my drip irrigation. I'd also call a trickle irrigation than a drip, but I just finished drilling 1,200 holes. Thank goodness. Yeah, this isn't about me. What can I talk about? What can I talk about today on today's show? What is there to talk about today? Besides more drought stuff, because I've sort of been going on a fair bit about that, which is quite relevant. It's going to happen in the Northern Hemisphere as well, just not in Australia and Africa, like it has for ages. Well, I've got no real direction today for the show. Might be heading towards a short show. Haven't done one of them for a while. I've got a drip irrigation to connect and dial in. Gosh, it's going to be good. I'll make a video of it. Because nobody's, from what I've seen, has made a clog-free drip irrigation system. So if it works, I'll make a video. Well, actually, I'll make a video even if it doesn't work, because you've got to put up your failures as well, because you still learn by those. The only thing that's going to fail me is pressure. So my pump, which is 500 watts, mightn't be big enough. I might need 1,000 watt to provide to the amount of 1,200 holes that I've got for the water to exit. But I look forward to trying it. We've got to adapt to in these situations of too much rain and too little rain, too much wind. Oh, here we go. Genetic Memory Farm says, talk more about your irrigation, meaning my irrigation. Makes for good content because you know it inside and out and have a good reason to deploy it. Many others in your community will learn something about adapting. Okay. Well, I currently use 7,500 litres, 2,000 gallons on a sprinkler to water the field twice a week. So I can't afford that in drought conditions, or I would be buying water. So my bore only produces 1,000 gallons a day. So I've got to work within those parameters in these conditions. So I'll quickly go through my water if I continue. So I thought I've got to change it. So the most efficient way is drip irrigation. But there's problems with drip irrigation. Let's go into it a bit then. Is the clogging of the emitters. That's the whole of that course virtually taught me that. I'll just get it up to the right area. Micro irrigation. No, I'm share now. Micro irrigation is here, and that's what I'm going to go into. Talking about emitters. The whole problem about doing these few plumbing courses that I've done, agricultural irrigation courses, is the emitters and getting the right pressure to supply to the furthest outlet. Let's have a look at some slides. Yeah, scheduling. I want to start from kind of the start. Methods of soil indicators. Oh, this is testing it. No, not going that far. We already know that it needs water. It's good getting into it here. So you've got to know your soil profile, and then you've got to know your depth of your roots, of the crop you're growing. So approximately, most of the time, they're in the top 4 inches, 100 mil. So therefore, because see the water to get down lower, it takes ages. So you've got to need more water to seep in and goes down. That's what this slide's getting at. Yeah, it's a good rule of thumb for us. I'm not going that heavily into it though, sorry. I'm also talking about once we get to the field. Well, we're already on the field with these techniques. The best way to test it out of all of these, that's a good one. I haven't tried that. They cost too much money. I'm yet to try them. The resistive sensors, they're made of gypsum. They're hooked up to a screen and it tells you how much moisture you got. I use these ones for years, the capacitive ones. They're cheap as, and they hook up to a screen and it's just got a number from 0 to 100. Well, you'll interpret that number when it gets down to low will be 20, whatever it says, and when it gets up to high. But the problem with that is they develop little pockets around as that plant grows, as the soil gets eaten up and that sort of stuff. So they're good rules guide. Get back to it. This had heaps of maths, this, and I'm not a fan of the math. Look at that, like that. Oh yeah, let's just do this, huh? There's a square root inside of a square root, and it's just great fun. Propel, you got to work out one of the irrigation efficiency. Water use efficiency is what I'm getting at in the whole of this. Irrigation efficiency is the ratio between the amount of water used to meet the consumptive use requirement of the crop plus the necessary to maintain a favourable salt balance in your crop root zone. Because when water evaporates, only the actual water particles evaporate, leaving all the salts, minerals, all those different elements that are inside of that water will remain, and that builds up the salt. You don't want that. It's only one of the big problems with irrigating with high ppm water. Efficiency for different irrigation systems. That's good to furrow, bridge and furrows. Yeah, I'm not going into that. Stick to the topic. Look at this. This is cool because I get taught the old school way as well. So if you can't afford a pump, no worries. You can make one. So here's the Archimedeal screw method. So you'll have a winder up here. Water will come to the bottom and you'll screw it up. No worries. Or a swing basket. You have it in the river like that and it's made of whatever it's made of and it retains a bit of water and you'll push it up, tip it over here, swing it back in, tip it back here. Here's another one, the counterpoise. Now they've got a counterbalance on the end. Or the Don method, we use your foot, get into it, back and forward like a seesaw. Or look, a chain pump. If you've got chains hanging around, you might be able to make a chain wheel one. Put it on a drum, wind her up, little tiny round things that go through a pipe. They pull the water up. And a water wheel. Righto. This is that salinity I was talking about before. Low, 150 on the ppm meter, 480 medium, 1400's high. Anything under 2000's all right. All right, should I say, depends if your crop is tolerable to high salinity. Yeah. But in general, you should be right. But over 2000, you're in a bit of strife. They reckon over 400, over 500, you can't drink. That's the EPA's guidelines. TDS is this division of it. It's 750, I think. You divide it by to get EC for TDS. It's the same readings, maths, some sodium absorption ratio. I'm not going to go into that. It's pretty good though. Please get back to the bloody field. Amendments. Right, I'm stopping this. I'm going to talk too much. Oh, look, right here. I was just about to stop it. The whole thing in this course, basically, this is the thing you want to pull out. Causes of clogging emitters. Well, I've eliminated this by not using an emitter. I'm not reading that. They clog for all sorts of reasons. You can imagine they've got little tiny holes. Anything will go into it. So pause it if you want to read that. Calcium's bad for it. And you've got microbes, they're called biofilm. Physical, chemical, and biological elements that clog up a system. So I went through this and I thought, wow, how can I eliminate this? You've got three types in its soil texture profile. You've got sand, silt, and clay. Sand, silt, and clay, they're sized. That's 0.001, 0.01, and that's above a millimetre. Anything below 2 mil. So I thought, hang on, if I can make the outlet above 2 mil, I can allow sand particles to go through, allowing a no clog system. That's all I devised, and I've put it in testing at the moment. Let's go to the field now. This bit, excellent. So you've got to have a good water source. Comes in your main line, then you run off sub-mains from that, and then your laterals will come off that, your side bits. That's the whole system. So what I've done, because it's top dollar to go and buy all this drip tape or drip line, so I made my own. I put holes in it. 2 inch, 1.5, 1 inch to 1/2 inch. So 50 mil to 25 mil to 13 mil is the size of my pipes. I've got hose ball valves in the end of each line, both sides. Therefore, I can turn each side on more or less, so I can tell how much pressure is needed just from that. That's it. Then only to get the right spacings and the right hole size, but that's it. People look at me weird and they're saying, what, you're doing your own? Yeah, and it seems to be pretty easy. That's it. So the only thing that's going to fail me is enough pressure. So, but I think I'll be right though. But it's still an experiment. Then I just expand it. See how many I can get on, see how many lengths I can put in. It's got my water use efficiency. It'll have reduced it by 90%. So I want to go from 7,000 litres to 700 litres every probably 2 days. You have to go off your wetting pattern in your soil. I think this goes into it, this course. That's pretty good. It does too. Here's your wetting pattern. So see, if you put water in the middle there, it's going to drip outwards, and it's going to, by surface tension, it'll absorb the water further and further. Capillary action pulls it out. So you've got three types of soil: your clay, your clay, your loamy soil, and your sandy soil. And your clay's wetting pattern will really flatten out a lot if you've got clay. If you've got loam, it'll really bowl up like a droplet. And sand, it goes elliptical. It goes really elongated. It goes through fast. So you've got to know your wetting pattern. So I've got sandy loam. So I can continue. So I've got a round ball. So that's why putting it in the middle, I'm just going to get it round. It's not going to disperse up or down. Key factor for this to work is your soil type, I reckon. But you can probably just adjust it. Here's some cool things that I learnt also. Placing your laterals to your crops. Do you need one per row? Do you need, where? Look at this one. 2 laterals to do 4 crops, 4 rows. That's rad. Anyway, I think this will do. That better have been bloody sharing. Oh, yeah, good. Oh, gee whiz. Your bore, meaning your well, yes. And its output is 1,000 litres. No, it's 4,000 gallons. It's 1,000 gallons a day for 4,000 litres. And what is your mineral content of your water and your well? Mineral content is 285 parts per million. So you are just using large emitters to, no, 3.5 millimeter hole. Yeah. What do you reckon? So by grabbing this, and there's a shortage too, not on fertilizer, not only on fertilizer, but on this agricultural pipe, because it turns out they need a lot of urea to make the piping. So their prices here has jumped up, and there's actually I had to hunt around to get some. So yes, things could be better, but it's good because once I have got that dialed in, so I just turn a tap on, I can control that tap by a program, just by buying some. There's a lot of systems out there to control a solenoid. So to control the solenoid, it's a auto magnet, and once it's energized, it either pulls the magnet in or out, so it shuts the gate on or off. So to control that, you need a timer, and there's heaps of different timers you can get. So I could make one, it's just a simple one, just by using that solenoid, or I could probably buy one, and they're hooked up through Bluetooth and Wi-Fi these days. So you can, wherever you are, you can just dial it in. I'm also going to get a water flow meter when I can afford it, a bit down the track, and that's tell me exactly the numbers that people are wanting to know, because at the moment I'm guessing my volume that I water at as well, just by tapping on the sides of my water tanks when they go down. GMF says, any chance of getting mineral build up at the 3.5 hole? Yes, definitely. Yes, always. Calcium is a putrid. Calcium carbonates build up really badly, and my water has limestone beneath it, so there has calcium minerals. So yes, you'll see it come out as white precipitation. The holes will look white. I'd expect that after a season. With 300 is rather low. 3,000, you'd expect it every week. Maybe you'd have to put a filter on. At present, I just use a basic 120 micron filter. So if mine was higher, I'd have to use a reverse osmosis and drip it into a tank and then suck it out of that tank. But I don't have to go there because mine is filtered through limestone beneath the earth, which is the pH is 7. And yeah, it's just below 300. Yeah, that answers the clogging, the whole question. And then there's going to be bark. There's going to be other things. There might be plastic still in there. So I've got to see. But if I turn it on full blast, it'll blow everything out, and then I'll dial it back. That's how I plan to dial it in. It's going to be good fun. Yeah, you like my turtles. Here you go. It's got a new tank for the turtle through the week. There it is, my long neck, eastern long neck. That's so cool. Yes, hello, buddy. It's so interactive with me. Always wants to hang out. Don't you? Yes, you do. You show your neck. The neck's the same size as its shell, which is 2 inches, 50 mil. Yes, and I got some azolla from my pond. See up the top. Little plants floating on the top. That's azolla, which you've got to be careful. They can double its biomass in 24 hours. So it depends on how good your water is and stuff like that. So you've got to watch that and take over. It has in my neighbour's ponds, but I'm going to harvest it for its nitrogen fixing, its nitrogen holding capacity. But yeah, this bloke's excellent. Well, it's not a bloke. You can't sex them until about 3 or 4 years. You've got to call it something like that. And one little guppy survived in here. It's like as if they're friends now. It's pretty cool. Oh, and this one. So that was my upgrade, $85 for that tank. And then this one's all my little critters. So you can see all them swimming around. They're waterman insects. And that's a feed for the turtle for in tough times when it's the pond's frozen or it's not snowing it or something. I can't get to it. So I've got to prepare for those times because I don't want the turtle to starve. Look after it. And anybody, yes, and I got plenty of them in my main turtle tank. I got turtle pond. I got 20 adults and about 20 babies. There's heaps. It's getting so much more. I'm enriching it with shrimp. Already put 200 in there. I'm going to go to the creek today and get some more. Put some in that tank. Yeah, good fun. Yeah, I get a lot of enjoyment. Oh no, buddy, hello. Yeah, you can say good day. It's getting so much bigger. Name is thumbnail. It was the size of my thumbnail 6 months ago. Yes, and you're big now, aren't you? Not a thumbnail anymore, but that's the name stuck because it's stuck. Oh, you go, a bit of a beach shot. Look out the window. There's the other window. Hello, goats. Hello. There you go. There's a little tour. Very good. Nope, not remove at this stage. Adorable turtle, says GMF. Yes. Smart making turtle snacks. I've got to prepare for the tough times. The animals can't suffer. If I've got to keep it captive, I have to look after it properly. Yeah. How's Cookie and Tilly doing, says GMF. My goats, they're doing good. Cookie's got her winter coat on. She's about 2 inches thick of fur. She's just feels like the softest thing you've ever felt in their life. They're the best mates in the world, fair dinkum. Miniature goats, but you have to get them bottle fed, and they're exactly the same as a dog, but they're quiet. Just excellent. Follow you around. So funny, random. That's the reason why I can't really travel much as my goats. I don't want to leave them. Even when I jump the fence, they start bleating. They get separation anxiety. It's funny. They start bleating and carrying on and stuff, and yeah, and you jump the fence back and they everything's all good again. They've actually had some visitors. They're wild goats who came to the fence. I suppose I can show you that. It's some pretty good viewing. They gave a kiss. The wild goats gave a kiss to my goats. I just thought, that's just so sweet. Made me pretty happy. Oh, there we go. I saw bacterial. I like the bacteria stuff. Oh, it's Imatic. Hello, Imatic. What's this? When you change your turtle tank, do you incorporate that into your plant feed? Yeah, it'd be a great idea to, yes, but I don't. Would be a decent natural supplement. It'd be excellent. It'd be high in nitrites, because that's what the piss comes out at. But you want nitrate, so you'll need. It's nitrobacter. Nitrobacter converts nitrite to nitrate. Would help. If I could, so I'll just add a bit of nitrobacter as an example to that solution, and it would be sweet. It'd be a perfect nitrogen feed. But there'd be also phosphorus and potassium in there. It'd be in smaller amounts. Actually, the phosphorus would be in a higher amount. You might get actually probably 20%, could be even your phosphorus, because there's a lot of your microbes that are in your poo, and they contain all the DNA that have all of the. I'm getting a bit sidetracked here. All the phosphorus, or then no worries on cross contamination. Because of the it says bacterial or viral crossovers. Well, yes, there could be cross contamination. If the turtle was in poor health, it might harbour up some problems. But because it's in good health, its poo that it puts out, the microbes are the numbers of good ones to bad ones are at a good amount. So that there's no chance, there is a chance, always a problem, because if you're giving something to a plant with a small or a weakened immune system, it could be susceptible. But in mine, no, it's all good. And I'm organic, so I build on things. So this is a building factor. So the microbes that are actually in there could be new, and they could be ones that are doing different things. Like we saw at the start of the show about how some of the microbe to exudate interactions with the plant exudates that it puts out, that they release different metabolites to help in various processes. So by having the new microbes in, that would help in that sense. I went off a bit on a tangent there, sorry. But I love the microbes. I was just looking at other courses for me to study on microbes, because I was speaking to a company overseas. They want me to do some study, some research for them. And I'm speaking to them about it, and I realised I've got a lot of knowledge in microbes. Get a load of this. I'll press play on the video. It was a wild goat that came over. My goats call in the wild goats, and it came over. And that's there. You can see the wild goat there. It's a male. And look at Tilly. She's trying to kiss it, and it wants to get in. Look at that. I had to turn the electric fence on. Look, shit, that's a big goat. Look at its horns. It's massive. But yeah, I was pretty stoked. They came over. Look at it trying to get in. It's cool, eh? Ah, kisses for my goat. How sweet. Until he didn't care. Cookie's just pissing. Oh, I tried to pull on the fence. There, the other goat's in the paddock behind it.
I turned the electric fence off. I think I'm going to turn it back on again. I don't want it to jump in. It's a bit bigger than the other ones, not as many.
I'm just standing at my window.
It's pretty big. I mean, not pretty big, but it's bigger. It's cool looking, though.
That'll do. I didn't really have much more action from that.
It just left. I didn't have to do.
Oh, I said it just left, so it's good timing. At the stage. Oh, that's how you do it. That one. No. That'll do. Yeah, there you go. GMF. That's the good interaction the other day. Thanks for asking, mate. Medic says nothing happens without microbes. Got that right. They are building blocks of how we eat and how we function, how everything happens. What's that axis? You'll know better than me. The gut to brain axis. I can't remember the name of it. I reckon it's microbial driven. He wants more than a pub visit from her, I'm guessing. Yes. Yeah, because she was bleating her blimmin' head off. Yes, that's what I thought. If she got in, I do want them to get pregnant, but I want them to get pregnant so that they are producing smaller miniature goats, which are 2 feet top of their backs. So if you buy one, mine are 57 and 58 centimeters to the tops of their backs. So you've got to be under 60. So you want to check that. Yes, GMF says, so are you composting the goat manure for feeding your red wigglers? Sure am. Sure am, mate. Yes, it's got a very good NPK, the goat manure. To be exact, it's got an NPK of 1.35 to 1:3, which is very high. Because cow is 0.3, 0.1, 0.2. Chook is 5 to 1. So goat is 1.3 to 1:3. And in that, it's also got high organic matter, which is the main thing. Because you want on your healthy soils to be fertile, they've got to be up to 3% of organic matter to be healthy. So if they're more than 3%, you're on a winner. You got compost. So my soils in the whole place now, 2 acres is nearly that, because they've been around fertilising the whole place, and I've got a lot of kangaroos that come in each night now that I try and help as well. I want them in. I like seeing all their manure around the yard. And the NPK of that is still to be determined. Can you believe it? I wrote to a scientist through the week and said, from your paper, do you suggest that the NPK is blah, blah, blah? And didn't reply. Because, of course, a study is just a study. They don't go outside of their studies. No, I'm not going to explain. Good on him for doing a study. So, yeah, and that thing at the start was pretty cool. You're talking about microbes. This was the slide at the start. The interplay between drought-induced plant root exudates down here and microbial metabolites. So the metabolites that microbes produce. How does they interact? Well, they give a fantastic drought resilience response. Through these processes, they produce the exudates, and then these metabolites give the plant more drought-adapted abilities. It's cool as. So by having turtle poo and all other different sources of it, different microbial sources, you can only win. Let it sort itself out, fight itself out in your substrate. That's what I do. Because you can have dominant microbes, of course. You don't want dominant pathogens. Good fun. Not the microbe stuff. Just got some trichoderma. And I'll show you this. This is the experiment coming up. So I'm playing off these two. I got these two microbes. The company swears by Azarium, so do lots. I said to them, the concoction you've made, Aspergillium will outperform it, and you've put other stuff in. So that's my experiment. The reason why I can confidently say that is look at the phytohormones that the Aspergillium releases. Auxin, gibberellic acid, and abscisic acid. So this is a fantastic one for drought. That's exactly works on a plant to induce drought responses. Abscisic acid directly response. So gibberellic acid swells fruit, and auxin helps with root development and other mediated processes in the plant, which there's heaps of them for auxin. It's produced in the shoot and goes polarly down to the roots. Auxin maxima. So yeah, indole abscisic acid. So anyway, that's going to be performed when the weather warms up. It's so fun. Microbes are the winner. Can't believe the amount of amazing results I've got from my little experience from using all these selection that I've got in front of you. I won't not stop talking about it. That'll do. We'll talk another hour about the microbes. But yeah, trichoderma, they're good for putting they're a pesticide, a biopesticide. So that's why you really want it. But this company, I don't want to talk about them. No, I shouldn't talk about them, actually. Everybody's still learning. I just put it as that. Everyone, education's learning, and there's plenty to research and find out in this field. That's why I love it. It's interesting. Just by using one different microbe, you can change the whole parameters of everything. It's just fantastic. And we've only discovered, what, 1% on the planet at the most? Wow. So by getting into the microbe field, and two microbes have only been around in discovery for a few hundred years since the discovery of the microscope. Better not get back into that far. Biomedic says, so many microbe rabbit holes. Yes. But you've got to see which ones work with each other, which ones are symbiotic, or which ones are beneficial, or which ones don't work with each other, pathogenic, or they prey on each other, non-symbiotic. You don't want those types of ones. There's plenty of them. So that's the hard bit, is trying to get communities of beneficials that are symbiotic. Not much work's been put into that yet. People are working with microbes and their actual potentials individually, but symbiotically it starts to get a bit hard. So that's where I like to get into that hard bits. And that's what people make these concoctions, which is one of those ones that I've been given. Try it out. All right, well, I can't wait to speak to your microbiologist and we can talk about this and that, because I've got some suggestions already. That's why they were very happy to deal with me, because they could see that I know what I'm talking about. Yeah. That's why I can confidently say from the different companies that I'm dealing with now and speaking to their microbiologists that my education is excellent. I'm very appraised. It's all sunken in, and I look forward to sharing it with others and other people asking questions so I can help them out. So I like all the people through this show. There's no real people with ulterior motives or stuff like that. There's all just nice people. So I enjoy helping those who need help. Oh, dry mouth. How do we fix dry mouth? I'll show you. Dry. How we do it. So dry mouth chemical beliefs. Artificial salivas, lubricants, and need to trigger the pathway. You might know this, Imatic. Trigger the pathway that releases moisture. So I've got a dry mouth at the moment. We know that certain things make the dry mouth. And so what we need to do, you find out what pathway it is, what chemical can do it, and then we see what chemical is similar in the terpene, flavonoid sector, and try that chemical on it. And if that's the same one, we can introduce that terpene into plants through breeding program quite easily, as you know, and bang, you've fixed cottonmouth. I love doing that stuff, but I'm not doing that anymore. I'm doing organic plant science. But that's how you'd fix cottonmouth, and you probably name it cottonmouth. It's so fun playing with individual molecules that do tasks and getting a bacteria or a fungus or something to produce that molecule, or a plant. It's well, it's a form of genetic engineering that you don't need a lab for. You need a lab to test it down the track, but if you're going to do breeding program and try and transfer the gene for cottonmouth, anyway, I'm off the track a bit. Put me back on track, people. Is that the end of the show? Did pretty good today. Talked about plant microbe responses with drought. Plant microbe. No, you call it drought. What was the word for it again? Oh, drought-induced. Oh, the interplay. Thank you. Interplay between plant and microbes for drought. That was cool. What else were we talking about there? Went on a bit of a tangent. Oh, irrigation, that's right. Irrigation. That was a big one. At the end, we're talking a bit about the turtle poo. Manure microbes. M-A-N-U-R-E. Yes. Manure microbes. Oh, GMF says, I think the real advantage is why you just said not needing a lab. Most of us have only practical applications and go go by, and thus we are citizen scientists like farmers before industrial revolution. Yes, you can, and making your own lab's pretty easy too. It's pretty easy, like tissue culture, as I say, you know, in a box, make it in lab conditions. GMF says microbes are far more dependable than chemical. Oh, once people realise that, they will sort of scratch their head. They'll be thinking, this can't be true, but it is true. GMF says fertilisers and pest management system. Microbes, they reproduce fertiliser. This is what I'm saying to my neighbour who runs that 16,000 acre farm. My microbes reproduce nitrogen as we speak right now. I got 5 different types. I said, you buy yours, which makes the shops very happy, because that's what the tax man and that's what everybody wants. Big marketing. They got to keep it everyone employed. It's a spin out how kind of easy it is, to be honest. From knowing what I know, it's like, what? I can get silica, mucilaginosis, transformed silica, a lot of buscular mycorrhizae, the fungi, work with phosphorus. There's phosphorus solubilizing bacteria, pain bacillus. What's that? My favourite, pain bacillus. Can't remember the last bit. Pain bacilli was renamed Gluconcidobacter disotropulus. No, polymixer. Pain bacilli polymixer is my favourite. Look what it does. Fixes nitrogen and solubilizes phosphorus. 1 microbe. Yeah. Or maybe we'll get some pain bacilli azotofixins. It produces nitrogen, solubilizes phosphorus, and mineralizes potassium. There's one that does it all. Just one. How about combining a few? Oh, bingo. GMF says yes. That iMedic says GMF. iMedic says practical experience is king, but to determine exact factors of influence, you need controlled lab tests. Yeah, but my test is going pretty well, and I don't do the lab tests. Just by combining, you've seen the list. I'm combining all of them and getting results that are unseasonable, that make annuals last for 3 years. Instead of it lasting 1 year, which it should die off, it lasts 3 years. I can get it to keep going through a few cycles, through microbe interactions. So the tests outside I understand that the labs are key to market it and to tell people exactly scientifically what we're doing to the numbers, but to test preliminary and to see if things work, it can be done, is a way to say it. Yeah. I'm not saying you're wrong though, but it can be done because I'm doing it. And I haven't got any lab tests. I can pay to get my soil sampled, and that would do. That would tell me, right, you've got a count of this much in it, of this much, this much, of this much, and that would go, cool. Then I'd exactly know by me applying it through irrigation, or by applying it through manure, or by applying it through multiplication in compost heaps, which one's been more beneficial. So I do understand that lab tests could that, but by doing it without lab tests, it's I'm still getting results. Yeah. Oh, no, Medic says, just to rule out confounding influences, especially outdoor growing. Yeah, to be more specific, you definitely need a lab test to know, righto, this is numbers happening in this. Oh no, well, your temperature's too low all the time. So this, the growth parameters of this bacillus species that you're using, this bacillus genre that you're using, isn't very good because it's too cold. So that's, and therefore the numbers aren't multiplying in your soil. So that's how I could get the answers, a lot of answers actually, but just to see what works through getting plant responses and plant adaptations is fascinating. Yeah, I love it. GMF says, living in Illinois, you can smell where corn and soybean farmers spray, even the spray indoors afterwards. Scary stuff. I wish they would just learn about microbes. Oh, mate, good luck. Hey, and on that too, I've got a solicitor now that's works for me, not works for me, but you pay to have them on side. Relation to spray, they've had a successful court interaction where they sued, they got a successful outcome through spray drift. So it's very much shown upon in the industry, and the governments don't like it. iMedic says it can be done, but quantitative versus qualitative results do matter. Yeah, yep, well said, mate. GMF says the farmers stay away from what they spray. Very distinctive smell. Yeah, well, they got the ppm on them, or they drive those tractors with air-conditioned booths in them, with all that HEPA filters already on them. They know what's going on. They don't want cancer or heart problems. So they know it's the problem is with the big farmers is they've got a system that works, that produces money, that they're still getting an income each year. Why would they change? Really, why should they change? They're earning their $500,000 a year. Why should they change? Because it helps the environment. Well, that doesn't really help them in the long run. So I can understand it from their perspective, and they're not invested into it as much as we are with the earth and with the microbes. We know where it all comes from and how it functions and cycles. So it is hard to get that approach across. It's nearly a generative thing. It's going to take a few generations to go through of their children, and then they're going to change their practices slowly. Yeah, because Australia's the same. I'm in such a minute. Oh, I met a organic farmer just over the hill. Oh, over the hill, 10 mile away, and he's called Psycho from everybody. Because he does organic farming. It's sad. And I said, why? And he said, people just that are not used to change, they're different, so they call him different. So yeah, but I know enough about the sector to know that those people that want to call me crazy, they're just uneducated. iMedic says here too. Really, where you are, lots of crop dusting around, either insecticide, fungicide, desiccant. It's putrid. Well, watch that, iMedic, because you can get a class action happening. This is your neighbours. You can get the government involved through their spray drifts. If you can taste it, that's not good. That means it's accessing where you are, but proving it now. So what you got to do is you got to film it before. So when they spray, go out and film your conifer leaves, and they'll be nice and green. And you're going to say, they just sprayed, look at them. What happens in the next 48 hours? You'll do a 24-hour and a 48-hour afterwards, and they'll be all going blotchy, and that's the material you need. Then you grab that leaf tissue, and the government in Australia will test it for free. You call the EPA. But in the States, I don't know how it works, but you grab some of that tissue, tests off to a lab for glyphosate, it comes back positive, you sue them successfully. That's the process to go about it. You also want to warn neighbours that you're in the area and you grow organically to keep in mind about your property. Then you've done your legal obligation to the community as well. That's how you get around it and win and sue their asses. Get a year's wage for it. Hello, Austin Grow 916. Tried every single germination method and still have no luck. It's your genetics, mate. I'm sorry. Genetics or temperature. What temperature you got your substrate at? How do you start your seeds? Austin Grows. Medic says, so much overspray and indirect application of the product, that's pathetic. GMF says, I think you have to sell them on efficiency and cost savings over buying fertilizer and pesticide. Sell them on getting better prices for their crops while spending less. It's you can get them by saying you don't have to fertilize. You'll have the same yields. You'll have healthier crops down the track. You'll be improving the soil and the atmosphere around it and your stock. That's how I get them. I got no comeback on that. Oh, Austin. 15 seeds, paper towel, soaked them in a shot of water, and 75. Yeah, it's genetics, buddy. They're dormant. Once a seed, every seed has a dormancy, and depends how long they've been under those conditions is to how easy it is to break the dormancy. So you've got a dormant genetics at the moment. What you've got to do is rupture it. So you've got, watch some of my videos on seed germination. You've got to, the tester is the outside of the seed coat. Get some pliers and gently crack it. It's just so you can see inside of it. Put it back into water. That'll imbibe water because it needs three things to germinate: the temperature, water, and oxygen. So by cracking the seed, you let water in and started that process with oxygen. Also, put your genetics inside of a paper towel or in a cup on top of water and with an apple. The apple releases ethylene, which is a dormancy breaker, which will help it go to germination and it'll produce a little radical coming out, which is the first little thing you see, the white tip. I hope that helps, mate. And if it doesn't help, get back to us. But yeah, you got dormant seeds, my friend, because you've done everything correctly and it hasn't worked. So that's why I can confidently say that. So now you've got to do some dormancy busting techniques and then you'll succeed. Oh, you're welcome, mate. No worries. You're quite polite. There you go. Medic says, in Canada, there's a tie between seed providers and fertilizer, weed and pest control companies, usually the same or similar branding. It's a shame, isn't it, mate? It's a real shame. Canada just did a good test on their glyphosate, and they found that it stayed in their substrates up to 10 years. That was a pretty good study. I went through a while ago because I really admire Canada. Lived there for 5 years and I'd like to live there again. GMF says, I like to vernalize seeds and put them into the fridge for a bit to simulate winter. Yes, good work, mate. They should always be stored in the fridge, and it helps with that. That helps with that process as well. Hello, Miss Lene says, g'day, Aussie. TC, hope you're doing well. Just talking a bit about seeds at the back end. Yeah, by storing them in you give the best genetic potential to hold the embryos live for the longest without making the dormancy hard to start it off. Can you believe that those pink DBs that you've all probably tried, there's still a 90% successful germination rate of them, and that's 6 years old? I'm not kidding. Because they were stored correctly, which is in very low humidity, in very low condition temperature, just above freezing. It's excellent. And yeah, by simulating that, vernalizing to stimulate the winter's in the seeds, it helps give a stronger flowering if you do that process. Imatic says it's I don't know what that means. Contracts. Links those items. Doesn't sound very good. Hope everyone's well, she says, yes. Miss Pink DB, yes. Pink DB with pukin is tasty, might I say. But Pink DB, yeah, because what is it? Glucan cross with Island Pink was pukin. No. Yeah, and Pink DB was Island Pink cross with Death Boa. Yeah, that's right. A seeds versus insecticide says imatics. Well, there's a big difference where their seeds are produced. If they're produced in a greenhouse, it'll be lacking the UV. So the plants can give different expressions from that. Also, if you buy off a producer who's produced seeds, who's grown with with insecticides, you'll find that you might have more pest problems. You will have more pest problems because those genetics won't be used to defending themselves. They'll be used to getting bottle fed by insecticides. So by sourcing organic outdoor growers seeds is the best because you're going to get genetic stability through microbes, which has been able to be enhanced through all the outdoor elements. But yet, who knows if they're not in the right area? You could get the genetics that have been in the cold areas and you're in the hot areas. So to activate those microbes, like from the mesophiles, which is what we want, you might be in the psychrophiles, which is down near 0 degrees Celsius. That's a bad example, but it is an example. Miss Lene says, I love the linalool pink had. Linalool, yes. Hello, Token Tom. How's it going? Miss Lene says, Kimbo also has linalool and dronol. Oh, very good. Need one with phytol, which is the the nerve blocker. Not many have those, and that was one of the elements which I am interested in working with. It's a bit of my old secrets. Holy mackerel, it's been 1:10. Been going on for a bit. I usually go for an hour. It seems a few people have joined at the end. Is there anything to talk about? Any questions? It was nice to help Austin Grows out earlier. That was pretty good. Anything else like to be discussed? Because, yeah, it's over an hour now, and I respect people's time. So I don't mind answering a few questions quickly. I like those fast questions, kind of like earlier. And then off the top of my head, I can just answer and feel confident in the answer. Gusty, such a gusty location, this one. Oh, righto. Well, thanks for moderating. That's your welcome, Token Tom. You seem polite, so I thought why not? Need more people to give questions and to share all this, share the streams out, because I enjoy helping folk. Because my plant science, soil science, and microbiology is pretty good in agricultural field. So I enjoy helping those out who have questions and who hear a lot of myths and stuff. I can just show the slides to shed some light on the topic. It's good fun. I enjoy it. And then from the more and I've still got more to study. I'm just looking at the moment for microbiology classes, more uni courses this semester, but they haven't put them out yet. Because I think that's my future, is agricultural microbiology. And then do the beneficial micro business, I think. That's the go. I'm finding the more I talk about it to people, the more that they're interested, and they haven't even heard about it. That microbes that can produce nitrogen and keep reproducing themselves ongoing. There's one of them. What's it called? Anyway, it produces 80% of the sugarcane's needs for sugarcane. The nitrogen of Azospirillum lipiferium comes to mind. Produces 80% of the sugarcane's needs, one microbe only for the nitrogen. How rad is that? So all you need is a handful of those types of microbes, and you've got your fertilizer taken care of forever, or for as long as they'll keep reproducing in your substrate. It's such a winning field. That's why organics is probably won right up until 200 years ago. But it fascinates me. I can get one microbe to do that. And then one trichoderma microbe can get rid of all these grubs with the muscadine disease. It's just, I just love it. Really, really, really do like it. Anyway, it's not about me, it's about everyone else. Let's wrap it up. Toke. That sounds lovely. Thank you for nice words. GMF says, I got to dip. Good night, peeps. Be safe. Yes, same here. iMedic says, 31 says, cheers. Good night, GMF. Good night to everyone. iMedic. Good night, Mr. May and GMF and Token Tom. Everybody else who's listening. Thanks for your ears, and how you can help me is you can get more people to interact with the stream. That's how you can help me. Not by money, I appreciate that, but we're poor, so no, try to get a few more interactions, and that would help me a lot, because I like the comments and can bounce off the comments. It's good to help people. So that's how you can help me. Thanks again. Press the thumbs up if you thought it was good, and I hope to see you next week. Same time. No, it's an hour earlier. An hour 13 earlier. Holy mackerel. Yes, before now. So thanks again. See you next week. Bye, everyone. Thanks, iMedic. Thanks, everyone. Good night. See ya. And good